Imagine not being able to breathe. Imagine getting up to walk across the room and having to stop because it is too physically exhausting for your body. Imagine having to go to the hospital and be put into a medically induced coma and hooked up to a machine that breathes for you because your body is physically unable to. Even worse, imagine not being able to get access to this life-saving piece of equipment because the hospital is already full of hundreds of other people who are just like you who just happened to get there first. You've probably heard of at least one similar scenario over the past 2 years in people who have contracted COVID-19. The novel coronavirus is an RNA virus originating in Wuhan, China. Though it typically produces flu-like symptoms, some cases, including those from newly emerging variants, can be far more severe, leaving countless people on ventilators, dealing with long-term illness, or even dead. Life changed overnight for almost everybody, leaving people to deal with new family problems or mental health crises with very little support. The world's response has been variable, to say the least, as governments moved quickly or, in some cases, slowly in responding to COVID-19. The citizens of these countries were meeting them with mixed responses. Though some people immediately fell in line with what was understood to be best practice, others had some different ideas about what was actually going on. Almost immediately, conspiratorial ideas about the severity of the virus, the true origin of the virus, or the purpose of all the government mandates and lockdowns started spreading like wildfire through the internet. And these ideas have delayed progress, but at the same time as they were taking hold, good things were happening. Scientists united in a way that was unprecedented to develop new diagnostic tests. Almost immediately, full steam ahead research efforts were started to develop a vaccine against the deadly virus. And they worked. The first ever mRNA vaccine was developed and approved for use by the public to fight COVID-19. This did not quiet the conspiracies, and as thousands of people flocked to get the new vaccine, others were less enthused. And I mean, who can blame them for being skeptical? After all, what even is mRNA? To begin understanding what mRNA is, we first must jump back in time to high school biology. Specifically, we have to understand how biological molecules like DNA, mRNA, and proteins function. Your body is made up entirely of cells. Each of these cells contains its own unique genetic information called DNA. And cells care a lot about protecting DNA, so it's housed in the nucleus, away from other cellular components. DNA is what makes you unique from all other people. It contains instructions on how to carry out life-sustaining processes. However, in order for these instructions to be carried out, a few other molecules are involved, namely mRNA and proteins. The proteins in a cell are a little bit different than what we normally think about when somebody mentions protein. They are not a food group, but rather they're a type of biomolecule that carry out nearly every life-sustaining process that occurs in our cells. The information encoded in DNA cannot be used to produce proteins directly. Instead, an important intermediate called mRNA is used. A certain molecule called RNA polymerase transcribes the information stored in DNA into the transient messenger molecule mRNA. mRNA is produced in the nucleus, where DNA is, but unlike DNA, it is able to leave and interact with the other components of the cell in the cytoplasm. Once mRNA enters the cytoplasm, it is translated into a protein by another cell machine called the ribosome. Through this process, cells are able to produce all of the items necessary for sustaining life. It may be helpful to think of this process in terms of music. In this case, DNA would be the idea for a song in a composer's head. mRNA would be the song that gets written down, and proteins would be the song being performed. Understanding this process is important in understanding how the new coronavirus vaccine works. Traditional vaccines rely on using dead, inactivated viruses or protein subunits of viruses. These components cannot make you sick. They are introduced through injection into your body, and once they're there, the cells of the immune system recognize that they are foreign and launch a response to destroy them. This response involves producing antibody proteins that bind to some component of the virus and mark it for destruction. Recognizing the particles as foreign also forms a memory of the pathogen in your body. When you come into contact with a live virus, it usually cannot infect your cells because of your immune system's memory. This mechanism is much different than that used by the new mRNA-based COVID-19 vaccine. To understand how the new vaccine works, it's important to first understand how the coronavirus hijacks your cells so that it can proliferate. Viruses like COVID-19 are enveloped in a lipid casing that protects its genetic material, which is RNA. Spike proteins on the surface of this virus help it gain entry into our cells. The spike protein binds to receptors on the surface of our body's cells, and this binding triggers a response that allows the virus to be brought inside of cells. Once in the cell, the coronavirus essentially hijacks the protein synthesis machinery we talked about earlier, and it uses those machines to produce more copies of itself. When these copies are fully formed, they are shipped out of the cell. The new copies of the coronavirus are able to enter and infect the surrounding cells, allowing the cycle to continue while spreading the virus throughout your body. Now that we understand how the coronavirus enters our cells, we can talk about how mRNA-based vaccines prevent entry and ultimately prevent illness. The Pfizer and Moderna vaccines are both mRNA-based. To refresh, mRNA is the transient messenger between DNA and proteins. mRNA in these vaccines is wrapped in an outer layer of lipid molecules that help the vaccine to be taken up by the body. Specifically, cells of the immune system called dendritic cells take up the mRNA. Once inside these cells, the mRNA is translated by the ribosomes. The mRNA encodes the spike protein, which is the part of the virus that helps COVID enter your cells and produce illness. On its own, the spike protein cannot cause an illness, and once the mRNA is translated into the spike protein, the protein is moved to the surface of the dendritic cells. Other immune system cells recognize this protein as foreign and begin to form antibodies against it. These antibodies aid in preventing infection in two ways. First, they compete with the receptors on the surface of the body cells to essentially neutralize the virus. Neutralization prevents entry of the virus into our cells. Second, they mark the virus particles as foreign and tell other immune cells to dispose of them so that they cannot produce an illness. At no point in the vaccination process does your body interact with dead or weakened coronavirus to elicit an immune response. Our DNA also remains untouched by the new mRNA introduced by the vaccine because the mRNA never even enters the nucleus where DNA is stored. So far, mRNA vaccines have proven safe and effective for every population monitored by the CDC. How can something that seemingly came out of nowhere already be touted as safe and effective? The answer is simple. This type of technology has been in development for decades. In fact, the first lipid-based delivery of mRNA into cells happened in 1978. But using mRNA as a therapeutic was just a dream back then. There were a lot of problems to work out, and the reason that nobody had ever heard of mRNA until 2020 was because of a few main ones. 1. mRNA is notoriously unstable. Cells do not try to keep around mRNA for very long. Rather, mRNA is supposed to serve as a transient intermediate between DNA and proteins. So figuring out how to get mRNA to stay around for long enough to be absorbed by your cells and translated was a huge challenge. And this problem was addressed by wrapping the mRNA in lipids. Lipids allowed for the mRNA to be more easily taken up by our cells. Figuring out how to ensure that mRNA didn't launch a dangerous immune response was another hurdle. Fortunately, scientists were able to chemically modify one of the subunits of mRNA that allowed it to hide from your immune system and be translated as any other native piece of mRNA. Finally, scientists have not always had the resources to produce mRNA in the laboratory-based way it is done now. Instead, they would isolate mRNA from animal sources. As you can imagine, this is very expensive and difficult, making it super impractical for large-scale production of things like vaccines. Developments in different biotechnologies in the past few decades have made it a reality. So even though it feels brand new, the precedent for mRNA-based therapeutics has existed for decades. In fact, clinical trials for mRNA vaccines for several different ailments, including cancer, have been ongoing for years without major safety issues being reported. Scientists have been running the marathon of mRNA vaccine development for decades, and we have only just witnessed the sprint to the end. If you're hearing me say all this and still feeling skeptical, maybe it would ease concerns for you to hear that scientists were the biggest critics and skeptics of mRNA therapies in the early days of their development. Very few people trusted the initial data suggesting that maybe one day mRNA could be used as a drug, and they had valid reasons to doubt its safety. These doubts, coupled with the logistical issues of producing mRNA, were huge barriers to the scientists that dedicated their careers to studying these processes. Catalin Carrico and Drew Weissman, the scientists largely credited for developing mRNA vaccines, struggled to get funding for their labs because of how outrageous these ideas seemed. Before the days of Pfizer and Moderna, Carrico actually owned a biotech company that eventually failed because nobody would buy into this idea. One key aspect of scientific skepticism is that it is not a fixed barrier. Enough data supporting a new idea will eventually convince scientists that it is worth pursuing. Enough people produce clear, reproducible results to support this new idea. Others read their results and decided that it was safe and worthwhile to pursue this new strategy, and these pursuits resulted in even stronger evidence for the technology, and eventually mRNA vaccines made their way to clinical trial. By all metrics, the clinical trials for the mRNA coronavirus vaccines have been very successful. The vaccine is effective at preventing illness, and the side effects are much more mild than severe cases of COVID-19. I hope this video has instilled a new appreciation for foundational biology in all of you. These concepts underlie the function of nearly all modern medicines that we use today. Understanding how they work is the way out of fear and distrust for science. I think you should get vaccinated, but I understand that's not going to convince most people. You don't have to listen to me though, but I hope you can listen to evidence and reason. Studies about the efficacy and safety of the COVID vaccine are free to access online. Tons of resources exist, including this video aimed at educating and dispelling misinformation around this topic. Skepticism is healthy, but only when it is flexible. Strong evidence in support of one idea is permission to believe in that idea. And standing in the way of therapeutic strategies that are consistently proven valid, safe, and effective by independent researchers only serves to delay the progress of science.